In this unit, we are talking about the characteristics of the various microorganisms. Chapter 5 talks about bacteria, and so we're going to go through and talk a little bit about some of their unique characteristics. Now, bacteria were the first microbes to develop on planet Earth. And as we go through this chapter, what you're going to learn, hopefully, is to appreciate the span of time in which bacteria existed, summarize the various forms of bacteria, some of their microscopic structures, describe the process that they reproduce, talk about how frequently they reproduce and what ramifications that can have, identify some of the environments which bacteria thrive, and recognize that they can grow in a wide range of environments. Outline several important groups of bacteria, and identify the importance of bacteria in some disease processes. Now, this chapter is mostly about just the characteristics of bacteria. We will talk about bacterial diseases toward the end of the semester, so don't have to worry too much about that at this point. Now, when we start talking about life on this planet, well, the Earth itself is about 4.5 billion years old. Now, life appeared in the form of bacteria, which are prokaryotic organisms, about 3.5 billion years ago. So, pretty much as soon as the planet cooled off enough to support life, we began to have these very simple organisms. And the fact is that these prokaryotic organisms, these bacteria, were the only inhabitants of the planet for about 2 billion years. We've only had more complex organisms in the last 1.5 billion. During that time, when only bacteria lived, the cyanobacteria developed. Now, this is a very important group of microorganisms because they are photosynthetic, which means that they can take light forms, plus take carbon dioxide out of the atmosphere, convert that carbon dioxide to a usable energy or food source, and in the process of that, generate oxygen. As a result, the cyanobacteria, when they developed, resulted in the oxygen content of the planet increasing from about 1% to about 21%. Earth had a high concentration of carbon dioxide in the planet prior to that and would not have supported life as we know it now. So it really paved the way for more complex life forms, the evolution of the cyanobacteria. Now, bacteria reproduce very rapidly. As a result, they can develop mutations very quickly that will allow them to adapt to changing environment. As a result, they have evolved to fill almost every environmental niche on the planet. They can be found 6 miles down at the bottom of the ocean, feeding off of heat vents. We find them at the top of the highest mountains where there is very little atmospheric pressure and it's very, very cold. We find them in just extremely hot desert climates. We find them on the North and South Pole where it is extremely frigid. The fact is, they are so numerous and so widespread that if you took just a single pinch of dirt between your fingers, you would have over 1 billion bacteria. Some of these are pathogens, but really a very small percentage. Most serve a role that is important in one way or another to the environment. Now, we've talked a little bit about the timeline here, and this is just a graphic to kind of give you a sense of the scale. 5 billion years ago, and then we have origin of Earth. We have some of the oldest rocks that were able to form as the planet cooled, and very shortly thereafter, we started having bacteria for a huge amount of time. It was just bacteria, but eventually, again, the cyanobacteria altered the environment in such a way that we could get more complex organisms like the eukaryotes to form. Here's 500 million years that we've seen animals. And in the lifespan of animals, humans have only been on the planet just a fraction of that time. Just some general characteristics of bacteria. They are much smaller than the eukaryotic cells. They are quite tiny. They are very simple cells. They don't have a lot of the organelles. Basically, they have a bit of DNA. They have some ribosomes, which are necessary for the production of proteins. They have transfer RNA, which is necessary to move some materials, but not any of the other organelles that you would see in the larger, more complex eukaryotic organisms. Their shapes, generally, there are three major shape forms. We see bacillus, which are these, cylindrical type structures. There are coccus or cocci, which means just little round structures. And then we have spiral shapes, and that includes spirillum and spirochetes that look like this. Now, there are other forms. There is sort of this comma-shaped one, which is sort of a modified bacillus called a vibrio. They're extremely rare shape of square, star, and triangle. And in our discussions, we're not even going to consider those forms. We are going to stick with bacillus, coccus, and spirillum, and spirochetes. We may refer to the vibrio. General morphology, they are very small, 0.5 to 20 micrometers. There is a very large sample, which was measured at 750 micrometers, or microns, I'm sorry, which is just about half the size of a period on a piece of paper. At that size, it is actually visible with the naked eye. Now, the cocci, we see those. That's a very common form, and they can arrange themselves in a number of ways. They are approximately half a micron in diameter, and they can come in twos, which are referred to as diplococci, or paired. They can come in tetrads, which are groups of four. You can get something called a sarcinia, which is a cubical cluster. And then we have streptococci, which are in chains, and staphylococci, which are in clusters like grapes. The shape and arrangement are important clues to identifying which organism you're dealing with. If you have a disease, by staining techniques, viewing the arrangements of these, you can narrow down what the disease may be just from those physical characteristics. Now, staining is an important part, first in just being able to see microorganisms, but it is important for identification too. Bacteria are very small, and they are practically translucent, transparent, so you don't see them very easily unless you stain. Admittedly, if you are staining them, you are killing them, so you're not going to see a live organism here. But again, you can still tell some stuff from shape, structure, things like that. There are a number of stains. There's simple stain, where you just use one dye, and an example of that is methylene blue. And you can just put that on there, and it gets absorbed into the cell walls, and you can look at those under the microscope. That doesn't really differentiate for you. What it will tell you is whether you have a cocci, or a spirillum, or a bacillus, but it doesn't tell you much else about the characteristics. To do that, one of the most used stains and most helpful stains that we have is the gram stain. And what we do is we use several dyes, and this helps us differentiate the cells. And what we end up with when we stain this way is we can have either gram-positive cells, which are going to have this sort of blue-purple color when they are stained, and gram-negative cells, which are going to have this sort of orange-red color when they are stained. Now, As I said before, gram staining is key in identifying the type of organisms that you're dealing with. And this also gives us some clues as to how to treat, say, some diseases. As some antibiotics are more effective against gram-positive, but less so on gram-negative microorganisms. Some antiseptics are less effective on gram-negative organisms. And it has to do with the cell wall structure that determines whether a cell is gram-negative or gram-positive. If we look at these characteristics, what we see is on the surface of cells, we have a cell wall. Not all microorganisms, but the ones that we're talking about right now, there is a cell wall which contains peptidoglycan and different amounts. It is a relatively thin layer in gram-negative microorganisms. It's relatively thick in gram-positives. And many of the antibiotics that we use prevent the formation of the peptidoglycan. So in gram-positive microorganisms, this is an important structural and protective mechanism on the cell. By preventing the building of the peptidoglycan, we can really destroy that cell wall and make that microorganism vulnerable. Now, inside, as we have with whole cells, we also have this plasma membrane, which has that fluid mosaic made of the phospholipid bilayer. It also contains proteins and some small carbohydrates, but this is the part that is intimately associated with the cytoplasm of the cell. This is an outer structure that adds additional strength and additional structure to the microorganisms. There are other things that exist on bacteria as well, some surface structures. In gram-negative bacteria, we will see a outer membrane, and lying between the cell membrane and this outer membrane is a material called the periplasm. And this consists of a number of enzymes. It has material that is being broken down for nutrients. It is an important part of these gram-negative bacteria. Some microorganisms have a glycocalyx, which allows adhesion and also helps them resist phagocytosis. It's like a big slimy layer on the outside, and it's not present in all bacteria. If it is a firmer gelatinous type substance, we think of it as a capsule. It's tight. If it's looser, we think of it as a slime layer. When we are drilling for oil, we often think that we are pulling up the remains of dead plant life and dead animal life. What we are more likely looking at is the collection of the slime layers of microorganisms that have pooled and have kind of, I guess, fermented over the millennia. Anyway, this is present in a lot of pathogenic microorganisms because it helps them stick to body parts. So this slime layer interferes with our immune system and makes this more likely to adhere to some part of our cells that and allow it to have access. We're going to stop right here and pick up on part 2 of the materials over chapter 5. Thank you.